Good morning, everyone. Today we are going to discuss a topic about restriction enzymes with a detailed note. So, restriction enzyme may also called as restriction endonuclease, which is a protein produced by the bacteria. So, it may also called as protein or enzymes. Here, you should know that all enzymes are called as protein, but not every other protein are called as enzymes. So, they may also call as protein or enzyme which is produced by the bacteria that cleaves DNA at a specific site. So, a protein produced by the bacteria that cleaves DNA at a specific site. So, which is naturally found in every bacteria. So, they have the capacity to recognize the specific base sequence on the DNA and have the ability to cut them on each strand. So hence they are called as molecular scissors. So let's see it with an example. So let's imagine it is a bacterial cell having the bacterial DNA, plasmid, and with lots of ligands, mitochondria, protein, enzymes. So here a small place occupied in the bacterial cell called restriction enzymes. So here the restriction enzyme are used to cut the human DNA genome, so hence you are called as molecular scissors, and it is applied in genetic engineering and for gene cloning, etc. etc. They restrict infection of bacteria by certain viruses, bacteriophage. So we all have the familiar with the word bacteriophage. So bacteriophage is a virus infected bacteria. So let us see it with an example. So let us consider an healthy bacteria without any disease having the restriction enzyme each in every cell organelles of the bacteria. So when an viral DNA is injected into or entered into the bacterial cell, it will be degraded by the restriction enzymes to protect the bacterial cell. Restriction enzyme plays a two major role. One is to restrict infection of bacteria by certain virus, and another role is to recognize specific base sequence on the DNA and to cut them. Each and every enzyme of restriction enzymes are named after the bacteria from which it was isolated. So the first letter should be capitalized on beginning with the bacterial genus name, and second and third letter should be with small cases named with the species name, and additional letters from the strain, so that may be rough strain or smooth strain like that. Let's see it with an example. Ecor 1. So here E stands for Escherichia and genus name, and CO small k stands for coli, specific species name, and R stands for RY13 and strain name, and 1 stands for first identify enzymes of E. coli. That's called Escherichia coli. So now let's see mechanism of the restriction enzyme. Restriction enzymes are specific endonuclease which cut both the backbone of the DNA. It recognizes specific short sequence. It will recognize a specific sequence on the DNA called as recognition sequence, which cleaves DNA on the particular recognizing sequence or away from the recognizing sequence. So it depends on the type of restriction enzyme you are using. Enzyme cut covalent phosphodiester bond between the nucleotide and hydrogen bond between the strands. So some restriction enzyme cut straight across the molecule of symmetrical axis producing the blunt end. And some restriction enzyme cut between the same two axes but away from the point of symmetry on the two strands, producing sticky ends. Let's see what is mean by blunt ends and sticky ends with an example. On the DNA sequence, if I want the particular position or a particular base from the DNA sequence, here I'm going to apply hint 3 and restriction enzyme to cleave the particular position in the DNA. So after cleavage, it resulted in the sticky ends that cut between the same two bases but away from the point of symmetry on two strands. So, which result in sticky ends. An example of producing sticky ends will include a restriction enzyme isosomers. Blunt ends are taken these sequence to explain. So, if I want the particular sequence from the DNA to be cut cleavage, are taken and restriction enzyme ECORV, in a restriction enzyme which digests the sequence and cleave into an end which will result in blunt end, which is a symmetrical axis producing a blunt end. So an example, neosomers, which is a restriction enzyme, will result in blunt ends.